Hi everyone, so today we're going to talk about bacteria. And one very important thing that I want to tell you at the beginning of today's lesson is that bacteria have many different shapes. There is the circular type, there is the rod shaped type, there is the spiral type, and bacteria can be good for us or they can be bad. Some species are good, some species are bad. This bad species that we probably heard of before is, or you guys probably heard of before, is E. coli and Salmonella. You probably also heard of a specific bacteria called Streptococcus that can cause sore throat, so people refer to as strep throat. If you're talking about a single bacteria, sorry, you were talking about a singular form of a bacteria, you can call it a bacterium. So Bacterium. When you're referring to many types of them, you call them bacteria. So a single one is referred to as a bacterium, many is bacteria. So how is a bacterium different from a virus? First of all, bacteria are complete cells. They can reproduce on their own, and they do not need to go inside of a living plant or animal cell for reproduction. Viruses, we know, however, from our past lesson, is that they need a cell to go into to replicate their genetic material. So viruses, they do not need a living cell to reproduce. Bacteria, they can cause damage by either growing inside of your cell, and by growing inside of your cell, they can damage your cell like that, or they can produce a chemical called toxin. We know that a virus, they burst your cell when they're ready to leave after they have reproduced. So bacteria, they don't burst your cell like that. They grow, and once they grow and they cannot fit in your cell bursts, and they also produce toxins that can actually, which are chemicals that can damage your cells. Um, Also, a very major important difference is that as much as they're viruses and they all cause um, diseases, bacteria, there are some of them that are actually beneficial, and we need them to survive. So that is a very big difference right there, is that as much as there are bad bacteria, there are also a group of very good bacteria that we depend every single day for survival. And during this lesson, you will learn more about that. So you're probably wondering, why did I say that bacteria are beneficial? We actually use bacteria on a daily basis. For example, all of our yogurts and our cheese, they come from bacteria because these good bacteria are used to make products that we use every single day. We also use them in brewing of alcohol and butter. We may make butter by using bacteria also. Bacteria, there is a number, there are thousands of types that are living inside of our digestive tract. And they're there because they help us to digest our food. Without those bacteria, we would not be able to digest all of our food. So they are very, very beneficial um, to us being able to digest our food. And when plants and animals die, their material need to be recycled. So bacteria help to decompose dead plants and animal and returning nutrients to the soil so other plants can use them and they can grow. So three benefits that I'm going to talk about, and there's so much more, but I just want to highlight some of them in this lesson, is that they can decompose dead plants and animal, returning nutrients to the soil. They help us in digestion, and they are used in industries for making cheese and butter and yogurt. Now let's look at harmful bacteria and what do they do that can cause us to be sick. Now, a harmful bacteria is referred to as a pathogen. Similarly, a harmful fungi, a harmful parasite, or a harmful virus is referred to as a pathogen. So there are four types of pathogen that we have been studying thus far. Um, Harmful bacteria also causes food spoilage. So that is something that we can also list as being harmful to us. Um, so there's a disease causing one that are called pathogen, and there are those that cause food spoilage. Among other things, there are other harmful bacteria. I just want to list two of them so you have a general idea. Today, we're going to only focus on pathogenic bacteria, those that cause diseases in our body. So, how do bacteria cause illness? Well, first of all, let's say, for example, this is our skin, and we have a cut or a scratch in our skin. This opens up um, an avenue for which bacteria can actually enter our cell, enter into our body. Now, once the bacteria enters into our body recognizes it. How do our body recognize it? Recognize it, sorry, because as I said before, microorganisms such as bacteria and virus, they have something on the surface that allows our body to recognize, and this something is called an antigen. So because of the antigen or the chemical that is present on the surface of bacteria, our white blood cells, which is a part of our immune system, will detect this. So now our helper cells will start to release antibodies. And I know right now you're like, wait a minute, isn't the same thing happen? Doesn't the same thing happen when a virus enter our body? Yes. Our immune system works very similar in getting rid of all pathogen. The helper cell will detect the antigen, it will release antibodies, and then it will send our killer cells or neutrophil or phagocytic cells also to either engulf or kill the pathogen by releasing chemical. Okay, so what happens if they do get into our cells? Well, if they do get into our cells, this is our nucleus, of course, they will start to grow and they will start to divide and they will produce more and more and more. And eventually, after they've used up all the nutrients of that cell, they will release or they'll burst through our cell to go to another cell, and the same thing will happen. Every time they damage a cell, they will actually cause the cell to lose its function and the cell cannot work properly. So they can do that, or they can produce a toxin or a poison, and those toxin can actually damage our cell wall. And once it damages our cell wall, our cell cannot work anymore. So bacteria can cause illness in two ways. One, they can multiply inside of our cell and damage the cell as they grow, or they can produce toxins that can damage our cell. We can actually prevent the spread of bacteria. We can prevent it by vaccination, proper hygiene, cooking food properly, washing our hands after using the bathroom, covering our mouth when we're coughing or sneezing, or using protection while having sex. We have talked a lot about vaccination as a preventative measure in combating diseases. I just want to recall some of the basics of vaccination and how it works. Basically, you are using the weakened or dead form of the pathogen. So if you are actually developing a vaccine for bacteria, you will use the dead or the weakened form of the bacteria. If you are developing a vaccine for virus, you will use the dead or weakened form of the virus. So once that happens, of course, your helper cell will produce antibodies, and then after it binds and it kills the virus or the bacteria, then what will happen is that you will start to make memory cell. And once those memory cells are there, they will remember the antigen of the pathogen, and then we'll have a better chance of killing them when they enter our body. That is the stronger version. So just as a recap of how vaccines work. So unlike viruses that does not have we don't have any type of cure for viral infection. We do have cure for most bacterial infection. And one thing that we use to cure bacterial infection is a drug called antibiotics. Now today you might notice antibiotics because they come in a capsule, and whenever you go to the doctor and they detect that you have a bacterial infection, they will probably give you an antibiotic. But what is an antibiotic and where does it come from? That is the important question we're going to answer today in today's lesson. So there was a scientist who were studying bacteria in his lab. So by studying these bacteria, he grows them in petri dishes. And here's a petri dish, an example of his petri dish, and all these yellow dots are actually colonies of bacteria that are growing. Eventually, he noticed something. He noticed that there was a fungi, that was growing in the center of his petri dish, and then all of the bacteria around that fungi was dying out. This is called the zone of inhibition, and all of these bacteria, after they were dying out, he came to a conclusion. Whatever this fungi was producing was killing the bacteria. And that was a breakthrough in medicine. Because if he could extract whatever this fungi was producing, he could give it to people whenever they have a bacterial infection to kill bacteria. And he did exactly that. The first type of first type of antibiotics was actually penicillin. Today there is so much more, and I'm going to discuss why there is so many types of antibiotics and why their effectiveness is being lost. Today, scientists who are doing research, they found many more antibiotics. Some of the familiar one you probably heard of is amoxicillin or ampicillin. Um, Basically, they use the same procedure as that scientist. By growing a bacteria in a petri dish and by using or dropping their chemicals, whatever it is, they're noticing that the zone around that the bacteria is dying. So if they're testing three, let's say one, two, three, four, five, six. If they're testing six, A, B, C, D, E, F. If they're testing six chemicals that can be used as an antibiotic, most likely I would choose F as an antibiotic because it most has a greater zone of inhibition, or most of the bacteria around it is dying out. However, there is a very big problem. So bacteria, they will grow, of course, and you can treat that by using antibiotics. And the antibiotics will go and this drug will actually kill the bacteria. However, in some cases, there are a few strains of bacteria that are resistant to the antibiotic. The bacteria, um, the antibiotic cannot work to kill these strains. The reason why is that some organisms have mutation. So bacteria that are mutated can survive even though you add antibiotics to them. These survived species will go and cause more infection and start to spread. Eventually, what will happen is that you will have something called an antibiotic resistance, where the penicillin or whatever antibiotic does not work anymore. So antibiotic does not work. And the reason why they don't work sometimes is because there are some species of bacteria that is resistant to it. And the reason why they're resistant is because of mutation. Let me draw a flow chart basically. I think a flow chart will help you to understand this concept a little bit better. So here we have our um, bacteria. Next, we're going to add to our bacteria antibiotics. So I'm going to label this antibiotics, antibiotics. The antibiotics will kill, sometimes it kills all of the bacteria. However, sometimes there are a few bacteria that can survive. The reason why these bacteria can survive is that they have a mutation. And what is a mutation? A mutation, for now, we're going to go with a very, very simple definition, is that a mutation is a change in their gene, and this change allows them to survive even though the antibiotic is trying to get rid of them. So they are resistant to that antibiotic. So because of a mutation, they will become resistant. And resistant means that the antibiotic will not work. Now, this being the case, scientists have lost a lot of antibiotics. So they start using a special type of antibiotic. Eventually, the mutation type of bacteria starts to grow, reproduce, start to spread, and the antibiotic could not work anymore. So they have to go search for a new one. And that will be used after a while, a couple of years, the same thing will happen. So there is no permanent type of antibiotic that has been discovered yet. Every single type of antibiotic that we have been using, um, the bacteria start becoming resistant to them after a few years. And the reason why is because these bacteria have mutated.